Hello, my name is Lisa Barnes and I'm a Senior Development Scientist here at Nu Skin. Today, I'm going to spend some time introducing our adaptogenic science, which shows the stress protection activity of adaptogenic botanicals and the exciting studies we've been performing. But first, we need to talk about stress and stressors to the skin. Typically, we only think of skin damage being caused by the sun through UV light. However, there are many other ways that the skin can be stressed and damaged. Both intrinsic and extrinsic sources can cause oxidative stress to the skin and can lead to aging. Extrinsic sources of oxidative stress include UVA, UVB, visible light, infrared, environmental pollution, and lifestyle factors. Lifestyle factors include diet, lack of sleep, etc. Intrinsic sources of oxidative stress include hormonal factors and chronological aging. Regardless of the source, oxidative stress impacts the integrity and look of skin. Some characteristic effects of skin stressors are dullness, inflammation, breakouts, acne, loss of moisture and barrier function, discoloration, and lines and wrinkles. Nu Skin took scientific insight from the plant world to help combat the effects of oxidative stress on the skin. We looked for ingredients that would confer stress resistance and promote cellular homeostasis that would, in turn, improve the skin's ability to adapt to stress through modulation of oxidative stress. Essentially, we looked for plants that adapted to their environments and looked to harness their adaptive properties to help the skin better adapt to the different stressors in life. We identified a blend of 5 adaptogenic plants that grow in extreme environments. These plants survive in environments including where it's extremely cold year-round, in the desert where it's hot and dry with little to no rainfall, in soil that is polluted, and lastly in high altitudes where soil nutrition is low. The blend includes Arctic root, marl root, Siberian ginseng, chaga mushroom, and the resurrection plant Selaginella lepidophylla extract. We chose this particular blend because each extract goes through an enzymatic process which breaks down the extracts to make them more bioavailable to the skin. The blend was also shown to increase heat shock protein expression and regulate inflammatory and glycation activity. Once we identified this adaptogenic blend, the next step was to confirm that they are still efficacious when combined with other ingredients found in different formula types. Initially, we performed multiple in vitro assays to confirm the adaptogenic blend's ability to impact key skin processes associated with stress and gene expression. The first in vitro study looked at a common skin process when responding to stress, specifically glycation leading to the formation of advanced glycation end products, or AGEs. AGEs are a marker of a linkage between proteins and sugars, resulting in brown spots, loss of elasticity, and photoaging. This study's objective was to look at 3 model formulas, a serum, a day cream, and a night mask that contained the adaptogenic blend, and see how they inhibited AGE formation to show an improved response to stress. The results showed significant inhibition of AGEs, with the night mask formula performing exceptionally well, inhibiting 42% of AGEs. All of the tested formulas performed as well or significantly better than data that had been previously generated on the blend itself, confirming the activity of this blend in formulas. The next in vitro assay performed was a gene expression PCR array that used intrinsic hormonal stress to induce a response. The same 3 model formulas were used, and overall we saw a downregulation of P53, which is a key stress gene marker, and downregulation of pro-inflammatory cytokines IL-1 and TNF-alpha. All our model formulas impacted these 3 genes, though there was some variation in the amount of downregulation of each gene based on the different formulas, which was expected. The results signal the regulation of the stress response, inflammation, and support for the immune response to overcome stress. Our final in vitro assay was another PCR array, but this time using extrinsic stressors, diesel particulate matter, and UVB exposure. We also included a 4th formula, an eye cream, in the 3 model formulas used in the previous studies. Overall, we saw a positive impact on the skin's defensive response to overcome external stressors. For the eye cream and the day cream, we saw an upregulation of the antioxidant gene HMOX1, some members of the heat shock protein family of genes, and an anti-apoptotic marker BCL2L1. The marker IL-1B was also downregulated. We observed an upregulation of a heat shock protein family gene for the serum and night mask, CRYAB, and downregulation of pro-inflammation markers IL-1B and NF-kappa B1A. Here is a summary of how key genes were impacted with samples pre-treated with the 4 model formulas compared to only being exposed to the stressors. You can see that when the model skin was exposed to stress alone, the skin's stress response was negatively impacted. However, the areas pre-treated with the topicals before stress induction showed a gene expression that indicated an improved stress response. Overall, the results confirmed our hypothesis regarding the adaptogenic blend having a positive effect on the skin and its response to stress. Even though we saw great results with our in vitro studies, we wanted to clinically test the blend's ability to work in model formulas to help the skin respond to stress. The in vivo protocol evaluated a serum, day cream, and night mask to determine if they decreased oxidative stress as measured by sunburn cells development following exposure to solar simulated radiation in healthy volunteers. The model formulas were applied to a site on the buttocks twice daily for 8 weeks. Each subject also had an untreated control site on the buttocks. After 8 weeks, the subjects returned to have the sites irradiated and then returned 24 hours later for evaluation. Dermospectrophotometer readings and photographs were obtained for all sites to determine if there was a reduction in redness between the control site and the treated site. Punch biopsies were then taken of each site and sent to a lab for analysis to look at the development of sunburned cells in the control versus the treated site. The 3 formulas achieved a successful reduction of the treated site's redness versus the control site, showing a reduction in oxidative stress when treating the skin with the model formulas containing the adaptogenic blend. Here are a couple visual examples of reducing the treated site's redness compared to its corresponding control site. Also, all 3 formulas had a positive effect on the reduction of oxidative stress as measured by the development of sunburn cells following exposure to UVB rays. The sites we treated with the model formulas with the adaptogenic blend had a lower sunburn cell count, showing that the formulas had an adaptogenic function. Of course, stress is caused by more than just UV exposure. It's a combination of intrinsic and extrinsic stressors. Nu Skin took insights from plants' adaptogenic abilities and scientifically tested a specific adaptogenic blend to see its impact on the skin to adapt to stress. Both the in vitro and in vivo testing results have shown that this adaptogenic blend positively impacts the skin's ability to adapt to its environment. Thank you so much for listening.